and I'll have a slide on that towards the end of my talk. And so Julie had emailed me a question about tilling, and then I said, hey, actually I'm going to be in India next week, and with everybody here and everybody behind the scenes that made this happen and made my visit happen, I thank you very much, and hopefully I will be able to explain things to you clearly today. So, am I okay? Yeah, usually too loud, so I apologize if I'm too loud. So what I'm going to tell you about is some work we're doing. So I'm working in Vienna, in Austria, and we're part of a joint program between the Food and Agriculture Organization, which is headquartered in Rome, Italy, and the IAEA, which is headquartered in Vienna. And really one of the mandates of our work is to support sustainable food security across the globe. And the FAO has a definition of food security. So I'll give you some background information, and I'm here, and everybody has heard all of these things before. So you've heard better versions of probably my introductory slides, but I'll go through them to set the stage for what we're doing and why we're doing it. So the FAO has a definition of food security that is a situation that exists when all people have access to sufficient, safe, and nutritious food that meets their dietary needs and food preferences for an active and healthy life. So this is a really well-rounded definition of food security, and more than just hunger. But we do have some serious issues we're facing, as you all know. So, you know, there's different estimates. One way to look at this is 1 in 8 people in the world suffer from chronic undernourishment. And there's a lot of pressures that affect food security. There's global climate change. I won't go through all of these, right? Arable land, population growth. And all of this really contributes to putting more pressure on agricultural production. And there's really a lot of approaches you could think of to improve food security. Food waste is a big deal in Europe. Right? A lot of food is wasted, and you could waste less food and feed more people with that. However, and I think I don't have to convince you guys that if you don't do plant genetics, you're really in trouble. Right? If you stop doing plant genetics, yields will not remain stable, they will drop. And so people have to keep focusing on plant breeding and what we can do to facilitate plant breeding globally. Now, of course, again, I'll go through these rather quickly. When we think about, you know, what can you do for plant breeding? Well, you can use variation for plant breeding, and variation within a species is one major resource for breeders. So these are just different crops, and you know all this already. And these variations are by and large variations, heritable variations in DNA sequence. Okay, so I would consider these mutations. And there's a definition of mutations that are changes in DNA sequences in a cell's genome. And there's a lot of ways those can happen naturally through things like errors in DNA replication. The act of copying DNA is error prone, and we have pretty good numbers on that by sequencing genomes now. And they also can be induced. So we can induce mutations. And basically, one of the reasons why you would induce mutations is that many crops have a narrow genetic base, or the alleles that you really want don't exist in nature. And there's no good way to get them into your genetic background. So we can induce mutations, and the first person that did this was Louis Stadler. So the very first person that induced mutations in anything was a guy named Herman Muller, working in Texas on Drosophila melanogaster. And in the late 1920s, really started this in the sort of the mid-1920s. You know, the history there is that people were rediscovering Mendel, and people started working on Drosophila in the early 1900s. And one of the reasons for that is if you've ever had a banana and you leave it in your apartment, it's covered in these little fruit flies, right? And you could grow millions of these things. And millions of organisms and millions of meiotic events are useful to find these rare natural mutations. But Muller didn't want to wait around for this, so he came up with a way of using ionizing radiation to affect sex-linked traits in Drosophila. And right about the same time, Stadler started doing this in cereals using X-rays. And really some nice work if you go back and read that, if you're a fan of plant genetics. Some of those early papers, 1929, looking at polyploid wheat is really pretty fantastic. And you can see Stadler's work, and this is, I think, in Columbia, Missouri. You can see this as the dawn of mutation breeding, what we call mutation breeding. And I would define that just using mutations to facilitate breeding, some sort of definition like that. And what you can do in mutation breeding is really treat sort of any kind of plant tissues from which you can grow a plant from. So seed, pollen, propagules, tissue culture material, cell cultures, etc. And you can do this a couple of different ways. You can treat with chemicals, you can treat with physical agents like ionizing radiation. Now, why would you do this, and why did Muller do this? Why do we do this today? Is that you can elicit changes orders of magnitude faster than happen in nature. So you really can create new variation very, very quickly. The first variety that was released was a tobacco variety in Indonesia in 1934. So you can see late 1920s, this idea comes to being with scientists, and by 1934 there's an approved tobacco variety growing, and this was a cigar leaf quality to it. So if we look today, the IAEA curates a database of officially released mutant varieties. This is a voluntary database, so it's up to the member states and the breeders to put their information in our database. We have over 3,200 officially released mutant plant varieties. This pie chart, sorry, standing there, this pie chart shows kind of a distribution of species. We see the top three being rice, wheat, and rice, barley, and wheat, and chrysanthemums is third there. So a lot of cereals populate a good percentage of the database, and I think there's a good reason for that, not only because they're important, but I'll talk about this a little bit later. About 224 plant species we have there now. And this is another way of looking at this, is how did people make these mutations? Well, most of those mutations were made with physical mutagens using gamma irradiation. So over half of these registered crop varieties were produced with gamma irradiation. And I'll get to that later too, because that might be important. Now I've got some examples of how this has been used. So the IAEA supports and funds research projects in many different countries to use induced mutations and what we would call a peaceful use of nuclear technologies to improve crops. And I'll just give you a few examples of these. And this is, if you're familiar with this, this is through the Department of Technical Cooperation in the IAEA, which you may hear about in the news sometime. So in Vietnam, one of the major successes in Vietnam has been in rice and salinity-tolerant rice that was created with gamma radiation. So since the 80s in Vietnam, they've released over 55 mutant varieties. The estimates that come from their government is that this has had an economic impact of over $2 billion US. 30% of the Mekong Delta is growing mutant rice, and they're improving that now. So they're continuing to add value to that through iron availability and also aromatic quality of their rice for export. So there's other, so they're winning awards for that locally. They estimate that over 8 million farmers have profited, and the economic impact of that, they estimate that the farmer's income has been raised by 30%. And we had last year given out for the first time plant breeding awards at the FAO IAEA. These were announced at the IAEA General Conference, and our Director General handed these out. And so they were one of the winners of that. I'll give you a couple more quickly. In Peru, looking at barley and amaranth, they've been able to find mutant varieties that grow above 3,500 meters, so high altitude in the Andes. And this is really important. That led to a 6-fold yield increase and over $6 million US annually to the income of the farmers. We have in India, there's examples in India. So India won a plant breeding award for over 42 crop varieties, including groundnut and high acreage coverage of groundnut. So these are just some examples. I have a lot of these, right, I could give you. But just to point out that in different crops, we have different examples of where inducing mutations can affect and increase the genetic variation. And that genetic variation can then be used to select useful phenotypes that farmers can use. So we like this, and we promote this, and for some of the following reasons. One, it's tested, it's proven, and it's robust. So since the 1930s, this has been used. There's over 3,200 varieties that are released. It's for the most part ubiquitously applicable. It's a hard thing to say, and I don't know why I wrote it that way. But the point is that, you know, if you can mutagenize something and you can do a little bit of genetics, you can do this. And there are some crops where that's hard, and I'll talk about that. There's no intellectual property with any of this, and it's not regulated. And that's important. We can mutagenize material and grow it anywhere in the world without any paperwork. And that's actually helpful to speed the work along. It doesn't cost very much, and as I said, it increases local economies. So now what I'm going to do is tell you the things that we do in my lab. And the plant breeding, it's not my lab, it's our lab, you know, a group of us. And we're called the Plant Breeding and Genetics Laboratory. And we are about 45 minutes south of Vienna. So if anybody's been to Vienna, Austria, and flown in at the airport, we are south of the airport by the Hungarian border. A little bit nice part of Lower Austria to work in. We do training, and I just had a little brief, so this is going to sound a lot like ICRISAC, because I just had a little brief tour and I was given this talk about all the things that you guys do, and we kind of do the same things, and I think the reason for that is there's smart things to do, right? You need to train people, right? And we have individual trainees and group trainees. We're a small lab. We're one little small lab. We are not a big group of people. And we train over 30 people a year. We run training courses in our lab. We also host fellows from anywhere from 3 months to 6 months. And we'll have over 15 to 20 fellows a year working with us, bringing their own material to try to develop methods. And then we also hold these training courses. So we've got Nawal here from Jordan and Estragita from Peru learning molecular mutation discovery techniques. We teach people how to work on seed in both vegetatively propagated species. So that's a big thing that we do in the lab. We also provide mutagenesis services. And this is Mirta Matajevic, who's running the Mutagenesis Services. And anybody is welcome to send material to us, and we can provide free of charge gamma radiation and X-ray radiation of their material. This is just a distribution of who uses our services. The top 3 are US, Germany, and the UK historically, but we see this shifting. So it's mostly countries that don't have their own irradiator or need help with optimization of their mutagenesis dose. So we follow the International Treaty on Plant Genetic Resources for Food and Agriculture, and you're all aware of that. This is administered or secretaried through the FAO. So we follow an SMTA for signatories of that treaty and those crops that fall under the multilateral system of that. We can do dosage optimization. So if you don't know anything about your crop but you want to try mutagenesis, you could send a seed, and Mirta will do some experiments and try to tell you what the best dose might be. And we're trying to do a better job at that, and I'll talk about that. We can work both with seed and vegetatively propagated crops, and we do about 40 requests per year, and this is all demand driven. So if somebody emails us, they want help, we do that, and we average about 40 per year. So that gives you a little idea about the demand. Now, one of the main things that we do is adaptive research and development and technology transfer rather. And I'll tell you some stories of this. So for the first is in direct support of these projects that the IAEA funds to different countries to improve their agriculture. So we in the lab try to think about what do they need, what can we give them, what kind of technologies can we give them, how can we help them be successful. That's number one. We also have, and don't be fooled by this acronym, we have what's called coordinated research projects. These are not CGCRPs. These are, and our name was first. So we've been using this acronym for years, but these are really, we get 10 people. So if we had a CRP in this room, we might say we want to work on chickpea, and we would get 10 people, 5 from developing countries, 5 from developed countries, sit around a room and come up with a research plan and just ask these upstream questions. What's going to be important in the next 5 or 10 years? What can we do to try to speed that along? What technologies, guidelines, what do we need to build to help people when they come ask for these projects for their agricultural improvement? And then another thing I'm working on, and I'll give you one example of that, is methods for labs with limited infrastructure. So how can we make basic methods more low cost and non-toxic? And this comes from visiting different labs and seeing the kind of problems people run into when they try to do molecular biology. So we really try to make that as simple and do it yourself as possible. So in our group, we look at phenotypes and we look at genotypes. I'm mostly thinking about the genotypes of plants. And when we think about how people do this, most of the work being done with mutation breeding is what I would call a forward genetic strategy. So we start with mutagenesis. In this case, it's rice seed, but you grow the material up to the first non-chimeric generation, and this would be the M2. And then at that point, it becomes appropriate to start phenotyping. And usually what people do is go to a direct varietal release. So many of these crops are just self-fertilized and directly released. Sometimes people want to know what the gene is. And we also do reverse genetics. So it's reverse genetics because it's the opposite of this. You start with the gene, and then you go for the phenotype. So you can make a large population, and I'll explain that in my next slide. But what I'm really interested in is where can we use molecular tools to improve these processes? So what tools are appropriate to facilitate cloning genes? How do we find mutations either cheaper or faster? It's kind of hard to do both at the same time, but when is it appropriate? Which technologies are appropriate in this stage? And then how can we use sort of genotyping methods to improve the efficiency of our mutagenesis? And I'll give some explanation for that in a second. I think my next slide will talk about reverse genetics. So as it was introduced, I've been for many years been working on something called tilling, and I didn't have much of a choice because when I was looking for a postdoc, I ran into Steve Hennicoff, and I was working on maize, and he explained to me this great reverse genetics technique for Arabidopsis, and then he told me it was called tilling, and I said, wow, that sounds a lot like my name. Maybe I should think about this a little bit more. It doesn't happen too often in your life. So it sounds like I named it, and the thing I always say to people, I meet some people, and they tell me, whoa, I thought you'd be much more arrogant, because, you know, who names something after themselves? And I think, oh my gosh, no, I didn't name it. But so Claire came up with this idea, and I know some of you are familiar with this, but let me just sort of give you a little bit of an idea. And this is what really excited me. So I'd worked on some transposon reverse genetics in maize, and I just saw the power of that. Like, I could find out what my gene is. I could mutate that. So I have an idea. This gene does this. I could mutate that, and then I could actually see what happens to the plant. And I had a chloroplast RNA splicing defect I found with a mu transposon, and I was like, wow, this is really cool. Then I met Claire by accident at a cancer research center working on Arabidopsis, and I decided to just talk to her because I wanted to know what she was doing with these plants. And she told me this, and it really amazed me. So she said, yeah, I make these mutagenized EMS populations of Arabidopsis, and then I grow them up to this M2. And then I do single seed descent, so I only have one. You know, each plant that I have in my population has different mutations. And then what I do is I take the tissue, and I make DNA from that, and then put that in the fridge. And that's stable for years. And then I've got seed, and that's stable for years. Now if I make a big enough population, and I have enough mutations in each plant, I can guarantee multiple mutations in any gene in the genome. So now I've got this library, right? Anytime I want a gene and I want to know what the gene does, I can find mutations in that gene. And it actually works, right? And I was really impressed by this. So, you know, the flow chart is you make these libraries of seed and DNA that really cover the entire genome. And you can do that because you're using something like EMS. And then there's a lot of work historically, and I won't go through all of that, but this part was a bit of a challenge to get that up and running and really high throughput. So we spent a lot of time figuring out the best way to find mutations. And that's not always the most expensive or the fastest way. And then what you do is, so you screen this library, you say, oh, that plant has that interesting mutation, and then you would go back to those seed and say, okay, did anything happen? And you can be very reductive in that. So it's a great functional genomics tool. So what are the advantages here? So you get, if you're using something like EMS, which causes point mutations, you can get a variety of different alleles. Right? You can knock a gene out. You can also get missense changes, and they can have varying effects on protein function. Mutations are by and large irreversible, so they're not going to go anywhere once you put them in. High density allows you to be efficient for small genes, even domains of proteins. You can target mutations in subregions of genes that you might be interested in. It's really like mutation breeding. It's the same idea. If you can mutate something and you can inherit those mutations, then you can do reverse genetics just as easily as you can do forward genetics. And you only need to phenotype the plants that you're interested in. So if you have a population of 6,000 and you find 10 of those having the mutation in the gene that you're interested in, you only have to look at those plants. So it can be efficient from a phenotypic standard point of view if you have a very difficult. And of course, it's non-transgenic, and that's not a political statement. That's a scientific fact there. So here's some examples. So over the years, this has been going on. We started doing this. Claire first published this in 2000. We opened the Arabidopsis Tilling Project up, I think, in 2001. And thousands of mutations have been gathered. And why that is important is it gives really good information on the effect of mutagens on plant genomes. And here you can see most things are EMS. I won't go through the whole table, but we learn about what happens when you mutagenize a genome, what kind of mutations you get, and what the density of those mutations are. And you can graph this out. So this is just some work that I did in my group back in Seattle, and then some work that Ann Slade did with wheat. and she was trained in our facility. So this data set is equivalent because it was all done using the same methodology. So we should be able to compare this. And what we saw over time, so this is the mutation density. So this maize pollen that was treated by Cliff Weil had a mutation density of 1 mutation every 500,000 base pairs. And then you move up, rice, about 1 mutation every 300,000 base pairs. So we can measure the mutation density and get an idea about what's going on. And what's interesting, one of the things that's interesting here is that we can see a big difference between diploids and polyploids. And I'll show you the banana data in a second. But really, as Stadler showed in 1929, genetically, genotypically, we see the same thing happening. That polyploid species are protected from mutagens because they have multiple copies of their genes. So that was really a nice thing that Ann's group at Arcadia Biosciences published in their polyploid week. This gives us a lot of predictive power. So we know how many mutations are in a population, we know how much work it will be, and we can provide some statistics on how many plants do I need to have a probability of getting alleles in the gene that I want. Mutagenesis is random, by and large. Mutations are randomly distributed across the genome, so you can be lucky or unlucky. So if you look at 2 genes, you might find 10 mutations in one, 2 mutations in another. That's just random statistics. There need not be any biology behind that, although I have to argue that a lot with people. But this gives us a lot of predictive power. And while many people started doing this looking simply for academic research, just functional genomics research studying gene function, people are starting to look into genes with traits of agronomic interest. So the group at Arcadia has found high amylose wheat. It's the first time that was done non-transgenically. Plant height, of course, is a big, I don't know how important it is for P, but it's a big green revolution pathway. Starch, group in Germany has improved starch for industrial use, and so on. So we see that you can make, of course you can, genes control phenotypes, and if you make mutations in genes and you alter gene function, you can have phenotypic consequences. So it's not surprising, but we're seeing it being done on that scale. Now, one of the things that I'm going to go back to this pie chart. One of the things that I really, when I started looking at this, I really asked the following question. A lot of cereals in this database, and of course cereals are very important. So of course countries would want to improve their cereal breeding, but there wasn't a lot of vegetatively propagated species. So we have projects on banana and cassava, which are staples for over 1 billion people in the world, and they're primarily vegetatively propagated. So there's been a lot of years of money going in to help people develop these projects, and not a lot coming out. And this is not a real fair comparison, and it's probably a little bit biased, but when I looked at this, I say, well, there's only 5 released varieties in our database, right? And maybe the database doesn't have everything, I don't know. But 14% of the world population depends on this, 0.16% populated in our database. Maybe we should just look at this a little bit more. So Joanna in my group, this is Joanna here, came up with this idea to look at bananas. We've been working on bananas for many years. Novak started some banana biotechnologies in Austria. So Austria is sort of historically a seat of some banana biotechnology developments. And we've been working on bananas for many years, but it's always puzzled me what goes on when you mutagenize a vegetatively propagated species, and how would this work? So Joanna took these are little cartoons of meristems with little leaf primordia there, if you can just imagine banana meristems. This is where the totipotent cells are, right? That divide to all the above, because the shoot apical meristem divides to all the above ground tissues. So she took that, soaked that in EMS, and then recovered those for 1 month, and then started to cut those meristems over and over again, because and then we took DNA and we did tilling reactions. And we were following this model, and I apologize, this is a little bit small. So here's the meristem, and all those little colored dots are to represent cells. When we mutagenize a population of cells, each cell will accumulate different mutations randomly. So we have this population, this heterogeneous population of cells, and in the middle here are the central zone cells that actually are totipotent and will divide. So the idea was that over time, you cut this in half and you reduce the genotypic complexity by half. And then you cut them again and you release it. And eventually, what you'd come out with is homogeneous material. And if you find a mutation, you could count the number of siblings in that line that have the mutation, and you could estimate when you got this job done. And that's important because it fairly time-consuming to do this. So if we could put a good time point on this, we could save people a lot of time and money. That's what we were thinking of. But what we found was something completely different. And we observed something which is known as diplantic selection, where by bathing these cells in a mutagen leads to variable competence. I mean, we didn't test this directly, but this is what we observed, and this is what we think. So if you bathe cells in mutagen, you will affect their cell division capabilities. Either the cells will die, or things will arrest in cell cycle. So you can have a situation where only one or several cells in the meristem survive in the central zone, and they populate. They will fill up the central zone, and they will populate that. So you can very quickly get something that is genotypically homogeneous, and what you'd see there is 100% segregation in your final line. And we took this out 6 generations, remember. And that's exactly what we saw, about 93%, in fact, not quite 100%. And we found the right kind of mutations in these populations, so that's just a graph of gene models and where the mutations are. 100% GC to AT, a density of 1 in 57 kilobases, which tells us when we look back at that graph, it's right above diploids and right below tetraploids. And these are triploid Cavendish bananas, IAEA bananas. So we think that we did a really good job with mutagenesis here. It falls exactly where we expect. That's why that graph, we like that graph. And it really tells us something about bananas and mutagenesis in bananas. And they're triploid, and these plants and other vegetatively propagated plants have accumulated deleterious mutations over time. So in many of these, in 4 out of 5 genes, they have a deleterious allele already. There's no way to get rid of this because they're sterile, parthenocarpic, and they don't undergo meiosis. So just continual mitotic propagation. So that tells us something interesting about the genetics and how we might use mutations to improve bananas. This was published in December of 2012. Okay, so now I want to move back to forward genetics. So I just told you a little story about reverse genetics. So let's go back to this chart. So I'm working at the IAEA. We're funding groups to use mutagenesis to go up this pipeline and release varieties, like I showed you in Vietnam and Peru and India. What happens then is people come to me through training courses with material, and they say to me, oh, now that I've done this, can you tell me which gene is mutated? Right? And that is an incredibly difficult thing to answer. So we have this problem. So most of these crops are self-fertilized. Nobody's done a cross. They've just let the plant self. They find it and they make a selection. They make another selection. The phenotype stabilizes and they release. And that's great. That doesn't cost much money, and you don't have to think too much about that. So it's not that technically challenging. But there's almost, so we don't know really what happens with any large data sets. What happens when you gamma irradiate material? There's a few publications, and some of them are contradictory. So we don't know what's happening there. We don't know any information on the genes driving these traits. So when I tell you that the rice is salinity tolerant, they're trying to find these genes. They don't know right away why those plants are salinity tolerant. And it gets very difficult to find that. That's the needle in a haystack kind of a question. And this knowledge would be very useful, not only for the biologist to understand genes and gene function and traits, but also you could develop perfect markers that then you could use to integrate these traits into other varieties. So one example is a technical cooperation project the IAEA supported for wheat stem rust race UG99, and I think probably you're all familiar with this. This is just a map from 2010 showing that UG99 was discovered in Uganda, probably in 1998, but reported in 99, and that it's been spreading around, right? And there's not a lot of resistance to UG99, and so this is threatening global wheat production in different areas. So in 2009, the IAEA created a project with this. This was spearheaded by Pierre Lagoda, my section head, involving 21 countries and 4 international institutions, where mutagenized material was sent to the University of Eldoret in Kenya as a screening hotspot. And the goal there was just forward mutation breeding. Can we generate variation in this material and then release varieties? And one example is actual genotypes, Kenyan genotypes. So I'll show you that now. So with Kenyan material, they sent the seed to our lab. So we did the service part of our work. We did the service. We gave them the mutagenized material. They grew it up. They did all the work for screening. And they followed these plants through many generations, and then they identified resistant material versus susceptible parents. So this is a mutant, that's the parent. They did that, then they sent people to our lab for training. We had training courses. We had, this is Ego Amos, he's there in my lab studying molecular biology, so we did the training part of our job. We did the service part, we did the training part. And they released 2 mutant varieties in 2014 in Kenya, which show resistance to UG99. So that is considered a success, and the project is mostly closing. And then they came to me and they said, okay, now you're the molecular biologist, tell me which mutations are in this genome. And I said to them, well, do you know how big the wheat genome is? It's big, billions and billions of base pairs. So this is hard, right? And I said to them, it's hard. And they said, no, you're the molecular biologist, you can tell us this. And I said, well, let's think about how we do this. And I'm going to be embarrassed. I have a little slide you can barely see, but this is my cartoon of a mapping population. So we said, you have to do some genetic process, people. So we got a guy, Hermann Bursmeyer, who's a wheat breeder in Austria, to come and give a lecture, and we came up with a guideline, and we said, yeah, it's going to take about 5 years, and you can do this. And they said, well, there's no more money. And how are we going to actually do this? So we decided, okay, how else could we do this? And we decided to try to adapt or at least develop a strategy for cloning by sequencing in wheat. So what I've done, and this is coming from my group and a lot of collaborations with people. So what I've done is Miriam Kinwa has sent me seeds so far from 5 of these resistant lines. There's about 12 or 13 resistant lines from different countries. So I'm collecting this material, and then I had to, so I'm worried about all this material coming from all over the world, and like there's probably some mistakes in that material. I mean, human beings, it's hard to do this without making some mistakes. So the first thing I did is I kind of came up with a mobile, I have an iPhone app that allows me to track my seed in my DNA. So I've got this little barcode system I adapted from a $5 application on iTunes. So I got that. And then we take this seed, and the first thing that we do is genotyping by sequencing. This is DART-seq. We're looking at about 20,000 SNPs randomly distributed across the wheat genome, and just asking the question, if you give me 2 seed packs and you say this is the same Kenyan variety, this is the mutant and this is the parent, they should be pretty genetically identical. So the first thing we're doing is validating all the material we get in through genotyping by sequencing, and Hermann's group in Boku in Austria is helping us with that analysis. And then I also want to ask the question, well, you give me a seed and you tell me it's resistant, is it really resistant? I don't know if it's resistant. How do I know that? So I trust people, but I also trust that we make mistakes, human beings. So what we've done now is we sent this to Tom Fetch's group in Canada, and they're doing a seedling phenotype validation. That's one way to look at the disease resistance. And I'm also sending this, and they just sent it last week. It finally cleared all of the import permits, but it's now with Rob Park's group in Australia. So he's going to do adult screening. So we're going to validate the genotype, validate the phenotype. So now I've got some material that I know, I hope I will have some material, that I know what it is. And now I can ask the question, how do I find that gene? And we're going with an exome capture approach. And I'll show you some data from exome capture. So what is this? And I heard this being done here this morning, so I probably don't have to explain this too much. But the point is, the short point is, genomes get big in plants, right? But the genes don't change that much. So if you just sequence the genes, and these are the coding regions, and the coding regions, yes, there are mutations that could be somewhere else in the genome, but mutations in genes are most likely to have an effect on protein and gene function. And so by focusing just on that, you save a little bit of time and effort in Arabidopsis, maybe a little less than 50%. You can reduce your sequencing load. But when you get up to wheat, you can't even imagine sequencing whole wheat genomes. I'm not going to sequence wheat. They're still sequencing the wheat genome by pulling out, Yaroslav Dozl's group, pulling out chromosomes through flow cytometry to sequence whole genomes. So we're not, sequence one chromosome that's the size of the rice genome. So we're not sequencing that. So we wanted to look into this sort of exome capture, kind of strategy. And people were doing this in wheat, George Zukowski's group with Luca Kamai. And so we worked with Luca to look at a couple other crops. And here's one more slide. So just to give you an idea, this sequencing the whole genome, and these little things are reads. And in here, you design liquid capture probes, right? And through hybridization, Watson-Crick kind of hybrid things, you can pull down only the sequences you want to sequence, and you get a little bit of junk. But for the most part, you can enrich the things that you choose. You could choose anything. You could do a promoter-based sequencing like this, but we were choosing the genes. We wanted to ask one question: does this work for gamma irradiation? Publications, all the pipelines were built for EMS. It can work. We wanted to know, does it work with gamma radiation? What do we get from gamma radiation? And we didn't know what we would get. We expected from the literature some small changes, maybe some big changes. So we thought we would give it a try. We tried 2 crops, gamma radiation of sorghum. These are 2 Sudanese genotypes that we had a fellow working in our lab from Sudan, and she brought some different material, so we gamma irradiated that. We took 15 plants from those 2 varieties, and Suleiman Bado and Owen Huynh and Joanna in my group worked through tissue culture mutagenesis of cassava. And I'm not going to go through that because we're still trying to sort that one out, but it's a little bit more complicated in growing this and following the genotypes around. But I'll just show you the sorghum data. So we recovered indels on this. And I should say that we did the work, and then that analysis, this is coming from Isabel Henry in Luca Kamai's group. So this is happening at UC Davis. Basically set the number of reads at 2 for a diploid, and then look for copy number variation. So this is a super scaffold, and this is chromosome 1. And in each case, you can see rather large, 575, 400,000 base pair deletions, so indels, in the genome. And you see that by seeing the copy number drop, right? And it's pretty straightforward. And in this case, all these little colors represent different dosages, controls. And what we see here is at 75 gray of gamma radiation, we've recovered in 2 siblings. We would expect there to be segregation. We see the recovery of these large deletions. And we see a range anywhere from 100,000 to 700,000. What we haven't seen so far is any small nucleotide variation, which is interesting to us. No SNPs, no small indels, only big stuff. We're doing more sequencing to really make sure about that, but it looks like, at least with sorghum, we're getting only large indels. And we found those in 4 of the 5 analyzed mutants in 1 sorghum line. So we had 5 mutants. In 4 of them, we find these indels. And I would say, wow, oh, it works. It's great. And how come I see as many in 75 as I see in 300? I would have thought if the dosage goes up, I would get more mutations, but I didn't see that. But then I would think, well, I didn't really find too many things, so maybe that's just an anomaly. But what's really weird is we took 2 Sudanese genotypes that are quite similar. We did the experiment ourselves. We grew them at the same time. We did the mutagenesis at the same time. And we didn't get any mutations in the other genotype. I mean, I don't know what to make about that. That's preliminary. But we're giving, the FAO-IAEA joint program, is providing mutagenesis services. We are providing material for people to do mutation breeding. And this right here makes me wonder if we need to think about applying some of these technologies early on to make sure that the material we're giving people, it has the highest number of mutations so that they can be successful. So this gives me hope that we can increase the success rate of mutation breeding by doing a better job. And I honestly don't know what's going on with 2 different genotypes. We have to repeat these experiments, but it's intriguing. And there's some more analysis that, that Isabel will do, so we're going to try to clean this up a little bit. And of course, we're putting more individuals in that. So now I want to tell you briefly about transferring technologies to other countries. So we are investing, of course, in these sort of higher end things like next-gen sequencing and exome capture and all this stuff, because we think those technologies will be quite powerful in helping people. But it's really not realistic to expect everybody in every country to buy a MiSeq and do these experiments or buy a HiSeq or whatever. It's really expensive and it can be kind of tough. But people still want to do some work, and there's good work to be done at a lower throughput. So we think about how do we transfer technologies to different countries, and I think about molecular biology. So different labs have varying infrastructure. Toxic waste is a really big deal. You would like to avoid that anywhere in the world, but especially in countries where there's no formal way to get rid of things like chloroform. That's potentially environmentally very bad. -80 freezers are annoying, right? I mean, if you have them, if they've ever broken down, they're a pain, but they also take a lot of electricity. And in places where you don't have constant power, like my colleagues in the DR Congo, you have to invest a lot in generators and things to keep these going. So do we really need those? Do we really need liquid nitrogen to collect tissue and to grind tissue? That's hard to get in some countries. So, there's these great molecular biology kits, but also people that don't know molecular biology need to practice, and that costs a lot of money. So you waste a lot of money teaching yourself how to do stuff. And I'm thinking about this from the DNA perspective. So the question is, how do we get good DNA to do things while avoiding most of this stuff? So my lab's been working on this. Low-cost methods that work in most labs. So this is Barnard Hoffman, who was replacing Joanna when she was having her baby. So these three guys worked together. So we started with tissue. And going back to literature, we did not invent anything. We read old papers, and we said, how do people used to do this? Well, our fellow from Sudan, Mayada, said, oh, I just put my tissue in silica. So let's test that. I don't freeze it. I don't do anything. I just stick it in silica, dry the tissue out in silica. Then how do we extract DNA? Well, you have a QIAGEN kit. You've got this shaking machine that breaks all the time. That's no good. Can we reverse engineer a QIAGEN kit using silica and chaotropic salts? Google it. There's a lot of examples of that. So we'll do that, and we'll adapt that protocol. And we'll use a vortexer. I've never seen one of these things break. So we tape our tubes to a vortexer, right? We put a little sand in the tube to grind the tissue with a couple metal balls. And we can get nice genomic DNA. And what we want to do is look for SNPs, do low-cost tilling and eco-tilling assays. And so how do we do that? Well, we can make our own enzyme. This is weeds I collected from my field in Austria. Just collect some weeds, and I can very quickly extract an enzyme from that. And I can use that to find SNPs. This is barley SNPs. This is a celery enzyme. This is a little bit better than just taking weeds from the field, but you can see the same activity. It's a single nucleotide polymorphism in a barley gene that we can see very nicely. So we've been able to do this. This is 1/10 the cost of a QIAGEN kit. No toxic chemicals. We use potassium iodide there. Everything can go down the sink. You can eat everything. Eat everything you want. Eat the weeds if you want, if they're not toxic. You can eat anything you want. The protocols, we've developed protocols in kits. So if anybody's interested in this, we'll give them a little bit of materials, mail it to them with a protocol. Protocols are being published. And we've trained over 80 scientists from 30 countries since 2013 on these methods. They are highly robust, which I'm really happy about. So we've published a little explanation of this. This Springer book for these methods will be out. It's online now, but the final download, it'll be open access. The final download wasn't there last week, so one of these weeks this will appear for free. And we've also adapted these methods. So how can we use these methods for other things? So one of the issues was when I was talking to people working on double haploids, they don't know if their plants are truly double haploid or not. And this is Jochen Kumlin at the IPK in Gattersleben. So we modified this, and we showed that this was a more efficient method to look for loss of heterozygosity compared to something like SSR markers. That's published, and we've since been adapting that with people. So with scientists in Ethiopia, we've adapted the method for TEF, and we're now looking at open pollinated sugar beet in Poland that they use to make double haploids. And that's pretty challenging to try to track the genotypes there. So we're sort of still working on that, but it's interesting. Now, it works. And this is this week. So these are photographs this week of us doing this at the University of Hyderabad. So Professor Rami Sharma has got this really nice 2-week training course, and the reason I'm in India is he invited me to come this week to teach them. And so I taught them this method. So this is the genomic DNA that these guys produce. Silica-dried tomato leaves. And this guy there that's a little bit degraded, that was fresh tissue. We just took fresh tissue and put it through. And we expected that should, everything during the lysis process, we should get some DNA degradation. So we see really nice things. And what's really cool about this week is we didn't think we had good potassium iodide. This is potassium iodide. So we used sodium iodide. I'd never tested that before. So multiple chaotropic buffers work. This is a tungsten carbide bead to grind the tissue. It costs €1 per bead. You can rewash them, but it's a pain. This is a steel bead that they bought from some bicycle shop in Hyderabad. They gave me like 1,000 of these beads to take in my suitcase. They cost nothing. Right? They can wash them and reuse them, but they cost nothing. So the one problem, I had a couple problems with my protocol. One was, well, not everybody can get the right kind of potassium iodide. And the other problem was I got these stupid beads that are so expensive. And this week we were able to do something new. And that was really cool. And here's us making enzymes. So this is really active enzyme. This is 0.1x, and you see the DNA is completely gone from a celery juice prep. And we developed a new method that uses benchtop centrifugation and little spin columns. And this enzyme is, per reaction, is 0.06 euro cents per reaction. You can do this really cheaply, no toxic chemicals, and these guys got it to work first time. So I'm really happy about that. And we're using this. We're actually transferring this to other countries, right? So we're not just doing this to publish papers and, that's great. We think the methods are useful. Here's some data from Babita Dusaruth in Mauritius looking at local banana varieties. So she was asking the question. She had a suspicion that the bananas that are grown in her country were not properly categorized. And there was some discrepancy in the phenotypic indicators that were being used. So she did low-cost banana ecotilling. So these are all the SNP variations in her bananas and also in reference accessions from the ITC. And she can see this. And then she was able to accurately characterize the genome composition of these two bananas. We've been taking low-cost DNA extraction, and the guys in Mauritius are great. We work with them quite closely. So we've now got this DNA extraction validated for tomato this week, jackfruit, chrysanthemum, roses, coffee, olive, Ceylon olive, lychee, mango, sorghum. So, I mean, it's DNA and it's tissue, but these things are really working. The only one that we've had problems with is bamboo, right? Somehow grinding the tissue. We haven't quite got good DNA from bamboo yet, but that's been working really quite nicely, and so we're quite happy about that. And I have a colleague, Mohammed Juhar in Syria, who's using this to genotype pathogens, which I'm really excited about. If anybody's interested in that, please talk to me. I'm trying to do this with tropical race 4 and fusarium and banana, but he started this. Syria is not an easy place to work, and he's been very successful. And this is C. sativus. This is a barley pathogen that produces xylanase genes. He's looking at the variation there, and that's looking really good. Cheap, easy to use. A lot of good data. You can just see the patterns here. This is ecotilling, looking at natural variation. And without even having to worry about what those sequences are, you can imagine giving this to somebody who has a collection of seeds and you want to know how homogeneous or heterogeneous are my seeds. Without spending any money, without doing a lot of high-tech anything, you could get a nice graphical picture just to say, oh, this one is different than that. How much, if this was a set of seeds that somebody thought was homogeneous, you could see it isn't, right? There's heterogeneity in that. So these are really cheap methods, and we think that they can be put to use for a number of different applications. Now, I'm working here with Ramesh Sharma. I met him in Vienna last year or something, and I was talking to him about expanding our tilling work. So I've got a little MySeq lab in my group, and I said, well, it'd be good to do tilling by sequencing. Luke is doing this, and these lycors are getting old. They don't make them anymore, and maybe we should do something with this. He says, well, I've got a tomato population for you. So he mailed this to me, and this is my experiment. So if you think the genomics people think this looks bad, I did the pipetting and the analysis. So what we weren't trying to be too careful with this. This is a first shot MySeq 3D pooled 2 times 300 paired end MySeq tilling by sequencing. So the first thing that we did, this is the data mapped against what I call the pseudogenome or pseudochromosome. So I developed just a genome of whatever I was sequencing. So the mapping's easier. This is the Illumina BWA map. And then this is one amplicon, and you get 2 times 300, but I set it at 310. So I get a little overlap there. So the spike tells me the middle of and you can see the read coverage goes all over the place. That's because I didn't care to normalize everything. I just wanted to see what happened and see where I could find mutations. We find mutations at the lowest reads. We lost one amplicon. We were debating yesterday whose mistake that was, and they wanted to say it was theirs, and I think it's my mistake. So I probably did something when I was suspending the lyophilized material. And then we just used the MySeq variant caller to give us data. And here's some example of this data. This is my analysis, and I made some mistakes, actually, which I learned yesterday. So the folks at the university were telling me how I did this a little bit wrong. Not really a bioinformatician, but I could do this, right? And that's kind of cool because I don't have all of this training, and yet I could go through this and very quickly within a week and come up with things that look like EMS-type mutations in the 3 dimensions of the pool. And we could pinpoint the tomato plant. And I'm going to give you an estimate. We have to go now and validate that these mutations are really in the population. So if we just say that the triplets are potential EMS mutations, we're at a mutation density of about 1 in 350,000. Again, going back to that graph that I have, that is right in the middle of where diploids should be, and so we're quite happy with that. Turns out that what Ramesh did to get the mutation density higher is he did 2 rounds of EMS mutagenesis. So my mistake here was well, I did a number of mistakes, but one of my mistakes was the biological one where I filtered everything out that repeated. And in fact, you expect some repetition of alleles in the population from the first round of EMS mutagenesis. So it's kind of a clever idea, but I didn't get that when I was doing this. But this sort of thing is working, and we think that this will be important. And the IAEA has just funded a project in Colombia where they're supporting a next generation sequencing. So the IAEA is starting to roll this out into their projects. We in the lab need to know what we're doing so we can give people good ideas about how they can do their work. I'm almost done. I'm almost done too, so that's good. So we're working in collaboration, and then hopefully we can find, if there's some interesting things that we could collaborate on, I would be happy to talk with you. Our group is small, and we can only survive through collaboration and through the generosity of good scientists. So we've got a lot of different projects going on. I know you work on different things in this audience. So we have near-infrared reflectance spectroscopy, some proteomics and metabolomics, working with Boku in Austria and also Wolfram Beckwerth's group at the University of Vienna. I'm working with Janice Zale at the University of Florida on a citrus greening project. We're hoping to get funding for that. That's a bacterial disease in citrus, so we're hoping to develop some tilling populations. We've done some grass p-tilling with Trevor Wang's group, Kathy Martin at the John Innes Center, and RevGen UK, and the populations coming from Bench Bio here in India. I've just started a project with tropical race 4, and this is an example of what we can kind of leverage if we're just sitting around and trying to think about what we could do. So tropical race 4 is really threatening all of the Cavendish bananas. There's really not a lot of resistance there, and there certainly isn't resistance in plantations. It's spreading around. I don't think it's been identified in India yet, but it's been identified in Africa. It's a big thing. The question is, will mutation breeding really work for this, right? And it's difficult. It's banana, vegetatively propagated. So we decided, well, before we do any kind of formal project, maybe we should investigate this a little bit. And I was talking with Altus Kolosian at Stellenbosch University in South Africa, and he talked to this company called Duroy. They donated 15,000 banana plants to our lab. They cost about €3 a piece to make. So they gave us a big donation. We got some money just out of our own pockets, and we found the Taiwan Biotechnology Research Institute that can do fusarium screening for tropical race 4. So what we're doing right now is we're taking all these bananas from South Africa, mutagenizing them, and sending them to Taiwan. And that's just an unstructured collaborative project. We're giving them a little bit of money to do this. We're also working on other things like the epigenomics of adaptation. So this is with Megan Snorberg's group at the Gregor Mendel Institute in Vienna. So a lot of different collaborations. And just to summarize then, so plant genetics I think will remain important. And I think that's not a tough argument to make because we have population growth, climate change, and things that are having a negative impact on food security. And we do need to continue to breed plants. We are going to mitigate that. Nucleotide variation, I hope you're convinced of this, is the major source of variability for the plant breeder, and mutations can be induced in most organisms at orders of magnitude than occur naturally. So we can create new diversity in crops, and mutation breeding has been widely successful, but we need to do more. We don't really understand this. I don't understand why one sorghum worked, why one didn't work. That's something I did wrong. That's something I did wrong. I need to learn that. And so I think we can do a better job, even with forward genetics, and reverse genetics allows us to leverage that. And so we need to improve traditional approaches. If people want to do DNA extraction in a country that has no chemical waste infrastructure, then we better give them some way to do that so they don't poison themselves, right? And if we have real good techniques that are new and innovative and modern that can save time and be fast, then we should also be thinking about those. So my lab is, we're trying to balance all of these things with a small group of people. And with that, I'll just say thanks. So this is the entire group. Stefan is our acting lab head. Joanna is directly in my group working on the banana stuff and a lot of other things in the rice metabolomics. I work closely with Bado on the cassava work, and Mirta runs the services. So not so many people. Pierre Lagoda is our section head, and then I probably have missed some people, but we have, as I said, a lot of collaborators I try to mention as I go along. And with that, I'll say thank you for your attention.
Thank you very much, Brad. And we have a few minutes. We have 5 minutes for some questions. Now, the camera there might be people who are awake, east coast of Africa, so I hope they enjoyed the seminar, and I think they can send questions. They can or they can't? No. Sorry, because we're not in the right room. This was a last-minute arrangement.
They could email me questions.
But anyway, people will be able to watch this seminar who missed it at a later date because it is recorded. So let's go with any questions that anyone has. And yes, Vincent?
Well, at the beginning, you made a survey of the number of variety that that have been originated from mutagenesis. Will you have the same sort of things in terms of surveys of existing population by species as a sort of repository where we can sort of contact people to get access to some material?
Yeah, what we have, so that's a good question, and that would be a nice thing to do. And sometimes when we talk about this database, people envision us in Austria having all of this germplasm and having all of these contacts with people. And really, we don't have the budget or the mandate to do that. And we argue about that quite a bit sometimes. But so this is a database where people enter their information. There is contact information. Some of the varieties date back decades. Where we can, we try to assist people, and we can assist with that sharing of germplasm. But that's somebody in the outside world entering a form and entering that with us. And we don't have any mechanism to force people, and we have no economists or anybody to go and do field studies. The FAO does have some of that, but in our joint program that doesn't exist. So we are just going with volunteers. But I would like to make that better, right? And imagine in the future, I mean, you have to probably go decades in the future, but imagine if we have these kind of sequencing technologies, and imagine if not only do you have this great variety, but we could say what happened, right? And one thing is nothing happened. It was sort of an introgression from something wild. It's a real mutation. It's a mutation in this gene. All of that information would be very valuable in a database like that. So I would like to see that, and I wish we had all of that material. But now for the UG99 project, I have to email people and say, please, 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 can you send some seed? Kind of difficult
sometimes. Reverse genetics, you gave example, like in one line you have hundreds of mutations. So if you are interested in particular gene, obviously you need to clean up the background. So how much work that will take to clean up those hundreds?
Well, if you look at you can actually calculate that, right? If you look at the density of mutations in your population, you can then estimate how many mutations do I have in a particular line. It will be a distribution, but it's an estimation. And then you can ask the question, looking at the genome, the size of the genome will matter there, what's the probability of having multiple deleterious alleles in my genome? And if you do a kind of calculation, this is a calculation that Luca did years ago, you might expect 2 or 3, right? So you've got a lot of silent mutations when you do something like EMS. More than half of the mutations are silent. They're in introns, they're completely silent mutations that don't affect the amino acid. So you have to do those kind of mathematical calculations. And this is a question that people have always asked. A geneticist would say, you don't really need to do anything. In fact, get 2 alleles, right? 2 mutants that have the mutation in the same gene. Just cross the 2 together. All the other alleles will be randomized, and most alleles will be recessive. So you can prove it in one go. When I talk to people that actually grow plants in the field, 4 back crosses, and you can calculate that as well. So people tend to think that 4 back crosses is enough, but there would be some work. In vegetatively propagated species, you can't do that, but you don't care, really. I mean, if you're going for the phenotype, publishing a paper becomes hard, right? But actually, if you can get a mutation and you can find the gene, and you can find multiple alleles, you can convince yourself that that gene is causative, and you can just go forward with the characterization. But that is an issue with the vegetatively propagated species.
I know that the wild species of banana have quite big seeds, so that's an alternative to doing the vegetative tilling.
Who's going to eat one of those?
No, I know, but if you're looking for
Yeah,
something.
and they don't always grow too well. We have so with this epigenomics thing, we're kind of asking about meiosis versus mitosis, and we're trying to get some of these seeds from Mauritius. They're not easy to work with either. Yeah, but for functional genomics, maybe you could do something with a diploid.
Yeah, it did interest me on your banana experiment. How many vegetative segments do you have? Are you keeping going in that experiment?
Well, we built. I went through that pretty fast because it's like a whole seminar. But we built a few thousand, population of a few thousand, and we being Joanna and a couple interns that I didn't mention that were in there for the summer, were just cutting like mad. And so what happened was we kept 768 of those plants to do this study. We kept those. We published a paper. We sent some of those to Nigeria to grow in the field, but we couldn't really meet. I mean, we don't have that kind of facility. We've got a small glass house where some bananas are growing, and we've kept about 50 lines in pots that we've also done. I didn't talk about that, but drought profiling and a microarray experiment to look at what's happening in the mutants when they're under drought stress.
Hi, it's a good talk. Like most of the examples using mutation reading or even killing actually coming in like disease resistance or like things like simply inherited traits. Like how they're applicable in, let's say, quantitatively inherited traits. Do you have some good examples?
Yeah, I mean, not off the top of my head, but if you can go to that database and search keywords. Now again, it's a database where nobody knows what happened to those plants, right? So we need more information. But if you look at that, and these, think about these gamma radiation, think of deleting 600,000 base pairs in a genome, right? That's a fairly profound effect. What we see is a lot of, I mean, there's drought. So we have temperature stress. So last month in my group, Benita from Mauritius was there with 5 tomato lines that were temperature resistant coming from gamma radiation. And again, we're doing the genetics now, and that's another thing that we will be looking for the genes to figure out what's going on. But if you just survey the database and you type in things like yield and abiotic stress, so you can understand biotic stress, an NDSLR, and you mutate that and it changes it. But there are examples of that. And the only thing I could say to that is throughout, if you're thinking about populations, natural populations, I mean, throughout evolution, the really severe alleles have been culled out. So there's sort of a subset of possible alleles that are floating around populations. And they're adaptive for many different traits, right? So there's a balance going on through the adaptation. And when we mutagenize, we're creating things that haven't been there before. So it is possible to make a dominant mutation in a pathway that could have a major effect, but you just wouldn't normally see that floating around. From the genetic standpoint, it doesn't surprise me, but I agree with you. It's easier to conceptualize the sort of normal single gene effects, but there's examples.